Good afternoon to the Aspen Ideas Health Day or Week. Thank you for joining us today, and I think it's a critical conversation regarding reinvigorating clinical trials. So my name is Stan Irk. I'm CEO of Novavax, and many of you may be familiar with our efforts with our COVID vaccine right now, but Novavax has been developing infectious disease vaccines for a long time in other areas, including not only the dozens of trials with COVID, but we've actually been the first company to develop an experimental vaccine in pregnant women, where we ran a trial in 4,000 pregnant women in 11 countries to show that RSV could be prevented in kids. So with that, as we discuss the vital role of ensuring that diverse populations are represented in future trials, it's my pleasure to introduce Alexis Madrigal, co-host of KQED's Forum, the leading talk show in the San Francisco Bay Area and contributing writer with The Atlantic. And Alexis will investigate how we can overhaul the clinical trial system, currently plagued by inefficiency and lack of diversity, and reach those who need next-generation treatments, and more quickly, efficiently, and equitably than those who need them more than ever. And join me in welcoming Alexis and today's esteemed panelists.
Yeah, thanks so much. The only other thing I wanted to add, somewhat germane to this, I was also the co-founder of the COVID tracking project, where I got to see a lot of data problems up close and personal, talking with states and federal government and other folks. And I've been writing about science, technology, and health for about 15 years now. So this is my field, and I'm so pleased to be here with you. Kind of quick frame up for this panel. You know, American clinical trials really have been one of the paragons of science for decades. They're rigorous, they're intense, and at the same time, the traditional approach has left out what people in the field often call, kind of hilariously to me, real-world data. And it's also left out a lot of people, specifically people in marginalized communities who have less access to the kinds of research institutions where trials have historically been done, or have their own histories of community trauma in relationship to clinical research. So what these changes, and the changes that we're going to hear about today, kind of add up to is a quiet revolution in what counts as evidence for assessing the safety and efficacy of treatments. And we could not have a better duo to help us understand the scale and speed of this change than this panel right here. Right to my left, Amy Abernathy is president of clinical research platforms at Alphabet's Verily. I know we all know this now, but Alphabet is sort of Google. It's a sister of Google.
Yes.
Verily is a sister of Google.
But it's
yes.
separate,
yes.
under the Alphabet umbrella.
Yes, under the Alphabet umbrella. That's right, that's right. And she has responsibility for the company's baseline program and other initiatives to support clinical trials and real-world evidence studies. Before joining Verily, she was Principal Deputy Commissioner and Acting Chief Information Officer of the U.S. Food and Drug Administration. To her left is Jonathan Jackson, who's the Executive Director of the Community Access Recruitment and Engagement Research Center at Massachusetts General and an Assistant Professor of Neurology at Harvard Medical School. CARE investigates the impact of diversity and inclusion on the quality of human subjects research and leverages deep community entrenchment to build trust and overcome barriers to clinical trial participation. So let's start pretty basic. For those who are not familiar with all the ins and outs of clinical trials, I'd love to have you, Amy, maybe begin by telling us about the traditional trial model and how it works.
So the traditional clinical trial model is fairly formulaic. We think about a series of phases of clinical trials. So phase 1 trials, really understanding safety of a new medical product like a drug, a device, a biologic, like a vaccine. And then in phase 2, now that we have established and understood safety, we start to say whether or not there is any evidence of efficacy. And then as those initial efficacy signals start to read out, and we also start to understand dosing and other features, then we move to phase 3, where really comparing the treatment to standard of care, whether that's placebo or other intervention. Then there is usually a high-risk decision like a regulatory approval, followed by what we would call the phase 4 setting, where systematic observation through more pragmatic studies to try and understand the performance of the product in more real-world settings. And that's the traditional way of thinking about clinical trials, but really over the last 25 years or so, that story has started to blur. So phase 1s and 2s have started to blur together, 2s and 3s. We've seen earlier regulatory approval, and I think we'll talk about this changing story, but that's the old paradigm.
Yeah. From your perspective, Dr. Jackson, how did this clinical trial model develop historically, and what were the sort of issues with the way that it developed?
Oh boy. So the historical clinical trials model was really designed, I'm sorry, I'm gonna have to kind of be as direct as possible. It was really designed to answer a narrow question with as much precision as possible. So you wanted to get rid of all of that other context and focus in on whether this particular biologic or whether this particular device directly addressed it, this very narrow target. And so you start off with these phase 1, which are now increasingly called first-in-human studies, to see, is it safe, and then does it kind of work, and then does it work the way that we want it to work? And so then in phase 4, you have this long-term monitoring where you start to say, maybe it's time to build in some of that context. But what we're learning is that context matters, and it matters even at the earliest stages of clinical trial design. So we have issues that I think are readily apparent, like a lack of representation, real problems with diversity. But we also know from the FDA's own research that 1 in 5 trials or 1 in 5 biologics that are approved by FDA have some sort of differential exposure or response on the basis of racial identity alone. And so, although the study didn't go into other aspects of socio-demographic identity, it's safe to say that maybe rural residents have a differential response, women might have a differential response, but we don't necessarily have a clear way of building in any way of analyzing that before the approval stage. So there's a huge inefficiency there. But then there are also questions about the way that we design clinical trials with that gold standard of the randomized controlled trial design. Where you're testing a therapy versus a placebo or standard of care. We also know that that is also not the most efficient way to do science. And then to get really nerdy, and I promise I'll shut up, there are increasing questions about the statistical rigor of the design that we choose. So we know just on the basis of selection bias alone, if you don't have a representative sample, it elides all 4, not just 1, but all 4 of the fundamental validities that underpin causal inference in science. So if you don't have a representative sample, you're not doing science. You're doing something, it's not science. So if we can start to build more sophisticated statistical models that take advantage of increasingly complex and flexible designs, and we put the patient at the front of that, then all of these differential things that we tend to put in opposition or tend to create tension around, they kind of fall into focus, and it allows us to do clinical trials that are diverse, representative, efficient, and patient-led all at the same time. So this is what the big idea is, and this is where we can go. Happy to talk about this in a little bit more detail, but I just wanted to set that landscape for you, that these things don't have to be in opposition.
Well, look, why haven't we had representative samples, given how important that is?
Okay, so I just rambled. I would love
No,
Amy
please.
Okay, all right.
We got time. I'm watching the clock. Yeah.
So the reason why we haven't had representative trials, if you look at the research literature and map out why people do or do not participate in clinical trials, there are effectively 11 reasons, and I'm not going to list all 11, but there are 11
Although it would be impressive if you could just tick them down.
I probably could, actually, but no, there are 11 big reasons why we don't have representative or diverse clinical trials. So some of them have to do with the way that we screen into trials, eligibility criteria. Some of it has to do with transportation and trust. Some of it has to do with basically not necessarily having great awareness. Even in the era of COVID-19, I know that many people here at the Aspen Ideas Festival still don't quite know what clinical research is. And so we have to recognize that clinical research is deeply weird, and we haven't done a great job of communicating that to the broader public. So, all of these reasons sort of create this environment where only the most privileged individuals will come into the clinical trial landscape as it is today. So we have the common rule, we have an entirely different research landscape sort of post-Tuskegee that still focuses on populations of convenience. So prior to the mid-1970s, those populations of convenience were people who looked like me. They were individuals who were disenfranchised and unlikely to speak out. After we implemented the Belmont Report and the Common Rule, those populations of convenience were still sought, But now, a population of convenience are rich, white, wealthy, upper class, overeducated, usually men who live on the east or west coast of the United States. If you're not all of those things at the same time, you are underrepresented in clinical trials the way that they currently are designed, which, as I mentioned earlier, elides a lot of the causal inference that we have in the design. So thinking about the people, thinking about the science, they go together in this really critical way, and we haven't done a good job about thinking about either of those, which makes it hard to have clinical trials that we can truly have faith in.
Can I add a couple of really tactical things? The first is that the reason that the clinical trial paradigm looks like it does is these are essentially experiments to understand whether or not treatments are adequately safe and effective to now be used at scale. And so there's a lot of anxiety about getting the experiment right. And so some of the controls around clinical trials, whether that's eligibility criteria or the deep requirements around collecting exactly the right data, are to make sure that the experiment is done correctly. And so that set of needs starts to exclude people. It starts to also create a bit of tunnel vision about doing the experiment as opposed to being representative. So that's one. The second is that if you're a person involved in a clinical trial in the traditional clinical trial paradigm, it's hard, right? You got to go to a distant clinic. You need your doctor to recognize that this is an opportunity for you and offer it up. You often need to take time off from work. You might need extra blood tests or even biopsies. It's just not an easy set of activities. And you start to put these things together, and then you add the third really critical angle to this, which is trust and why. Why me would I want to be involved in a clinical trial? And the history that we've got in clinical trials, whether that's Tuskegee or knowing that this is hard, has made this a system that's been hard to access for many. And so as we start to think through all of those three things, the design of the experiments, the burden of the process, and the building of the trust, those are three things we've got the opportunity to now solve and get right.
Yeah. I want to talk a little bit about what I want to call regulatory infrastructure around some of these changes. And in particular, I've heard you talk about the importance when you had, both your FDA hat on and your Verily hat on, Dr. Abernathy, the 21st Century Cures Act. For people who aren't familiar with that legislation, which I think that's okay, tell us a little bit about why that is kind of foundational for some of the changes that might come.
Wow, 21st Century Cures is an important piece of legislation that was signed in December of 2016. And as is, in the title, the whole point is to try and figure out how do we accelerate cures for the afflictions in our health. And practically speaking, one of the fundamental elements here was to figure out how do we speed up the process of understanding what works for whom and when, including how do we get the data that we need, how do we design and execute better studies, and then also how do regulators think about studies that have a sort of different set of features. In that sort of list of categories, there's some very basic things that are in 21st Century Cures that are pretty interesting. So first, 21st Century Cures has helped to set the framework, for example, of more interoperable electronic health record data that can be leveraged up and potentially used in evidence generation in the clinical trial landscape. Another aspect of 21st Century Cures was saying, you know what, we need to be smarter about putting the patient at the center and ensuring that patient-reported concerns are incorporated into the design of clinical trials and our understanding of medical products. Another aspect was say, you know what, we need to learn how to use novel clinical trial capabilities such as adaptive clinical trial designs and more efficient designs. You sort of talked before about some of the mathematical approaches that we might use. So let's learn how to use those, and let's get smarter about the endpoints that we need in clinical trials so that we can be more efficient. So sort of this idea towards efficient design. And then the last piece I'd highlight that was important in 21st Century Cures was how do we get smarter about using all available data? And you sort of highlighted this kind of funny construct of real-world data. We'll probably come back to that. But how can we start to incorporate data that comes from real-world sources, like the electronic health record, administrative claims, real-world settings, such as more generalizable clinical trials, into the decision-making and regulatory thinking around clinical trials? Now, 21st Century Cures did not say, FDA, you must tell us how you're going to do these things. It said, FDA, you must think about this and then provide guidance. And FDA came back and said, yes, we will start to provide guidance. And that we've seen those guidances come out through the end of 2021, really in line with those areas. One of the pieces that really wasn't as well surfaced in 21st Century Cures was issues around diversity and inclusion. There were some, but not enough, and we see that now starting to show up in newer legislation and really starting to figure out how do we do this better in the future.
Yeah. Do you think legislation is necessary, or do you think that the scientific imperatives that you're discussing, now that they sort of have been foregrounded, companies and the FDA will work it out even without legislation?
To be honest, I think legislation can at best sort of set a floor for this. But based on what I've seen coming out of industry and academia, we're all about to do an absolutely terrible job of diversity. And that's because we don't know what diversity is. We haven't operationalized it. We haven't connected it to the other primary aims of clinical trials, which is to generate evidence that eventually scales up to help us understand the efficacy of a particular biologic or device on some sort of health outcome. So right now we're treating these like they're two completely problems in opposition. And so, of course, we're going to sort of maintain fidelity for the problem that we understand and that we know, and we're going to try to incorporate these little pieces of diversity every now and again. And I'll just be honest, it's going to be a rough ride. And I think what I actually anticipate is there's going to be a significant backlash to this diversity trend in the next few years when we can avoid it by thinking more about what I'll call inclusion rather than diversity. Because that sort of sends you in slightly different directions. By thinking about diversity, you're like, oh, I've got to have one of every type, but then how do I do like a sub-analysis when I have like an N of 4? Like what's the point of looking at these tiny groups in the context of these large research questions? But when you start to ask the question from a place of inclusion, you start to say, oh, I see. A lack of diversity means that there's this enormous selection bias occurring. There's probably a measurement bias that's occurring. There's maybe a statistical bias that's occurring that's impacting the quality of my results. So I'm going to make these different design decisions in my research study, and as a consequence, one of the symptoms of those improved, more high fidelity kinds of scientific decisions, we're going to see, as a consequence, better representation instead of just looking for diversity. So you want to look for the individuals who are maybe starting at phase 2 and onward, representative of the burden of the disease or burden of the risk of the disease that you're looking at. And so that gives you a very natural set of goals to aim for without necessarily trying to kind of wrestle in these ideas related to diversity that are, really resist any sort of operationalization because they are ultimately right now being seen as moral mandates rather than scientific ones. And if we can make it a scientific endeavor, then of course it will improve the quality of our science.
Yeah. We've gotten a kind of decent sense of where we've been, maybe kind of signaled to people where we're going, but I think we want to address kind of this trans-pandemic moment. I can't say post-pandemic, so I'm just going trans-pandemic. We're still in it clearly,
Peri-pandemic.
But something's happening that's a little bit different. What changes did the sort of imperatives of speed around COVID-19 bring to the clinical trial world for people who haven't been following?
So, it's really important to think about what we've learned in the pandemic with respect to clinical research. I reflect on the COVID vaccines, and the story in COVID vaccines, I think, is a microcosm, or macrocosm maybe, but really a story that also summarizes a lot of what's been happening over the last 20 years with the shifts in clinical trials. So we end up with a FDA and other regulatory agency around the world decision based on clinical trials with fewer numbers of patients, randomized, but fewer numbers of patients and shorter duration. And then what we see is that after that regulatory decision, initially authorization, later approval, then in the post-authorization setting, really all of us needing to truly understand how these vaccines work in our everyday life. Duration of effectiveness, safety concerns, including, for example, myocarditis. What about children? Can you mix and match vaccines if you need a booster? All of these questions really came from real-world data and other clinical studies that happened after the regulatory decision. And that shift in clinical trials and evidence generation happened in COVID-19 really and started to show us how we might start thinking about data, how we might start thinking about clinical trial designs, and how we might start thinking about longitudinal evaluation of medical products across their life cycle in new ways. It also brought to the fore critical issues of representation, access to clinical trials, and access to being able to participate in the overall system.
Am I right that a lot of the data that was used, sort of post-authorization at least, wasn't from the US, right? It's the Israeli data, it's like the British data. Why is that?
So it's quite interesting. So I spent a fair amount of time recently in Israel, seeing what does it look like in Israel as it relates to being able to generate these data sets. And in the United States, we have a hard time bringing data together across systems. Even when we went from paper records to electronic health records, we still have pretty unique data systems in different places.
That's one way to put it. Yeah.
Tried to say this nicely, that aren't easily brought together. And then even if we can bring it together, it's very hard to get data into the same format and readily analyzable. Now, we can do that, and we've shown we can do it in various ways, but needing to do it on the fly in the context of servicing the management of the pandemic was very hard. Whereas you look at, for example, a place like Israel, and a lot of the data came from a health system called Clalit. They represent about 50%, 40 to 50% of all the people in Israel. They're all in the same electronic health record. The data were already basically in one common database ready to go, and they'd already learned how to work with those data sets, so they knew the trials and tribulations of where the missteps might be. And they were able to now partner with, that point was Pfizer, to now plan those studies as Pfizer was now getting their initial vaccine clinical trials done. I think it's a huge teaching about what is possible, and it also helps us understand if we're going to do that, we need to deal with issues as it relates to just the data and how do you get those things together, but also how do you think about the public conversation about how data sets might be used and how we might do that science.
Yeah. Dr. Jason, how about you? From your perspective, this COVID period, how did it affect the issues you're?
Yeah, so from my perspective, I was embedded deep in an academic research hospital, Mass General, and what was really remarkable was seeing the totality of the workforce shift. Every single lab, every single individual that were working on 10,000 different problems really came together almost overnight to start working on one common challenge. And I think that was one of the many great accelerators and catalysts that allowed us to design and then bring into trials these vaccines for COVID, and then to be able to test them with rapidity. What was really remarkable from my perspective was that for the first time there was an ability to question these systems. So as Amy has highlighted, there was this old model of the way that you do clinical research, and maybe you might be able to wiggle a little bit here or a little bit there, but you couldn't really change a whole lot about it. With COVID-19, and so the downside of that is that on average, that pipeline takes about 10 years to get through like early stage design all the way through like a completed phase 3 moving into phase 4 clinical trial. 10 years at best, right? And so now we're starting to say, well, maybe can we blur some of these phases? Maybe can we change the way that we do our design? Maybe we can change the way that we have our data safety and monitoring boards look at the data. Maybe we can change our questions about blinding. Maybe we can change the way that we think about outcomes and start to think about them in a way that makes sense, not just for us as research scientists, but start to incorporate some of that patient perspective in terms of what is meaningful in terms of their quality of life. So COVID allowed us not just to ask those questions one at a time, but to ask them all at once. And what we realized is, it doesn't actually have to be this way. It doesn't have to be this slow model that is deeply inefficient, that is really reliant on heavily federated data systems that are not interoperable in the United States. We can start to cut down on all these inefficiencies and do so, and especially in the case of the Moderna trial, quite dramatically. We can also do that while paying attention to issues of representation. because historically, all of these things are usually thought of as in opposition to one another. If you do this, you can't do that. You can't have an efficient trial and achieve diversity. That was the rule of law before COVID-19, is that you had to give up one or the other. And so as we saw, if you look at the Moderna data, they break it down week by week in terms of that diverse representation. And you could see rather dramatically in the late summer of 2020, you see, that we're getting mostly white people and not too many people of color. They basically stopped enrolling certain populations and turned it around in a 1-week time span. And then for the rest of the duration of that trial, they saw enrollment that was a little bit more, not quite, but a little bit more representative of the individuals who were being diagnosed and suffering from COVID-19. So it's possible to do.
How did they do that?
So what they did was a few different things. So they slowed enrollment at sites that didn't necessarily have access to those people of color. But then they also, I actually consulted on the flagship trial in Boston. And so what they ended up doing was this massive public-facing campaign. These are things that we've been saying that you need to do in clinical trials for years, but nobody's listened. They said that we don't have the time or the money, or there's not the will. But by going directly and appealing to the public, by going to primary care and talking to individuals there, by slowing enrollment at sites that can't necessarily enroll representatively, so again, not diversely, because that's not like the question that we're talking about. We're talking about people who are likely, or people who have gotten COVID-19. These are the individuals that we're looking for. And so by prioritizing and giving additional resources and training and having like this really top-down approach, it was possible to turn around in a very short amount of time. And the truth is, I've done much smaller clinical trials and research studies, and it turns out that the number one thing that you can do to get more diversity is to ask some diverse people to participate. Ask them nicely. This is not just there's a new National Academies report that came out last month that says exactly the same thing. The largest determinant of participation for diverse individuals is just being asked. So Moderna, basically asked, and it's astonishing that it took a trial of that sort of kind of priority or prestige to really put that question to the test. But it's not rocket science. We talk about how difficult it is to find a way to cure Alzheimer's disease or cancer or certain kinds of heart disease. Diversity, representation, it is not that complex of a challenge. There are so many barriers that we can overcome by simply treating people like people.
Yeah.
Can I mention two additional, and I
Sure.
think that extend from this, two additional important, and we'll call them innovations, I would say accelerations that happened in the pandemic. The first is the recovery trial in the UK.
Yeah.
So this study basically, and it's still ongoing, it's called a platform trial. So one group of individuals who don't get any new treatment for COVID, and now comparing each new successive treatment against that common control. Mm-hmm. And in this particular study, every patient in, I think, 130 participating centers in the UK, when they came in, a short form was filled out in addition to participant consent. And then from that point forward, they were exposed to one new intervention or the standard, so that might be remdesivir or dexamethasone, et cetera. And essentially, this study, which has now enrolled over 40,000 people in the UK, has really been one of the places that we've learned the most about initial treatments for COVID. This was where the dexamethasone data came from. This is where some of the hydroxychloroquine data came from, for example. And so really being able to see a platform trial run at scale is one of the things that was very, very visible in the context of COVID. The second thing that was an innovation or acceleration in COVID was the use of solutions that meet patients where they are. Electronic consenting, patient-reported outcome solutions, leveraging, for example, an app on the phone, the ability to use video visits. What happened was we had these solutions really in our clinical trials armamentarium before, but now suddenly people couldn't leave their homes. So how do you participate in a clinical trial if you can't leave your home? We need to now leverage these solutions for so-called decentralized clinical trials. And what happened in the context of the pandemic was we suddenly saw how they were working, and we suddenly saw how to embed them into our clinical trials landscape. We also learned they don't work for everyone. Some people want to call on the phone, some people want to see their doctor at the clinic, so we still need to figure out how we put all these pieces together, but we did see what this looks like working at scale.
So, I think 2 questions on the decentralized clinical trials. One, are those going to stick around through time? And 2, does that make it easier or harder to create these representative samples that we're talking about?
So the first one is easy to answer. Decentralized and virtual trials are here to stay, for sure. And I think that there is a really, really interesting new emergent focus on how to make these as efficient and flexible as possible so that we can maintain the gains that Amy, I think, highlighted. In terms of whether that will guarantee a representative sample, the answer is no. If you look, you don't have to be like a great student of history. I'm a better neuroscientist than I am a historian, but if you look at historical advances in technology, historical advances in medicine, they tend to disproportionately harm the most vulnerable and marginalized individuals. So we have to assume that with the acceleration of decentralized and virtualized trials, which is at the intersection of technology and medicine, we can't assume that it will inherently guarantee better representation, better equity for these populations that we're trying to consider. We need to design that intentionally. And it turns out that if you design from the perspective of patient-led trials, if you design from the perspective of equity-centric research, then yes, you can leverage decentralized and virtualized trials to achieve a representative sample. But you can't do it the other way around. If the converse is not true, a decentralized trial will not necessarily get you that representation that you're looking for. And I think, again, there are also lots of really recent examples, not necessarily in the clinical trial space, but in sort of the kind of the observational research phase. So for example, I did a lot of work with the Michael J. Fox Foundation, who had a virtualized trial called Fox Insight that they ran for many years. And so they said, this will be great. You don't have to see, a neurology subspecialist in a brick and mortar institution. Sometimes there's only 1 or 2 of these in a state in the United States. So we're going to put this online and we're going to get a representative sample. Turns out that the sample that they got of 25,000 individuals was 90% white. The median annual income was $104,000, and a third of individuals had a doctoral degree, either a PhD or an MD. Now, unless you're saying Parkinson's disease disproportionately affects really wealthy, overeducated white people, we cannot assume that that a virtualized trial will inherently give you that kind of diversity. So there are levers that you can pull that may help, but it's not a guarantee.
Amy, it seemed like you wanted to say something about whether virtual trials, decentralized trials, were here to stay. You were like,
I think that there are a couple of really critical components. And to say, first of all, at the outset, I sincerely believe we must incorporate virtual elements into our clinical trials. But I think there are 3 things for us to think about. The first is that as we have built those elements into our clinical trials in the context of the pandemic, we need to go back to first principles of what clinical trials are all about understanding safety and effectiveness of medical products. And we still don't know fully whether or not decentralized elements embedded in our clinical trials lead to the level of data quality and patient safety, as well as patient representation that's going to be needed in order to be able to make confident decisions about medical products in the future. So unless we go back and do that step of cross-checking what happened to data quality when we had truly virtual capabilities in the context of the pandemic, we're never going to know the answer to that question. And I can tell you that regulators are like, need to know that before I'm going to truly feel confident in this. So that's the first thing I would say. Second thing I would say is I'm an oncologist. So my frame of reference is tough drugs, tough treatments, often with lots of adverse effects, and oftentimes requiring patients to get biopsies and other interventions. And those trials are often hard to conduct completely decentralized, right? Especially if we're worried about new learnings about toxic effects. And so there are some trials that will be really very easily conducted in a decentralized frame, but many of them actually still need a site-based component. So then that takes me to my last point, which is I think we've created a false dichotomy. The false dichotomy is centralized versus decentralized in the same way we've created a false dichotomy of clinical trials data and real-world data. And in fact, the world that we're moving to is decentralized elements, when appropriate, for the study design that is needed, where we know we're going to get the right quality of data to answer the question credibly and where people can be kept safe.
Yeah. Just want to let everyone know to get your questions ready. We're going to get some Q&A here in a second. Last question for me before we go to the audience, though, is to think about the biggest challenges to creating this better clinical trial system, right? I mean, there's big reports, 20, 30 clinical trials from the National Academy. There's all these different ways that people have been looking at this. But from your particular part of the ecosystem, like what's the single biggest or the couple biggest challenges to improving our clinical trials?
Yeah, I'll start. I would say, and this is something that we haven't quite touched on, ultimately, the way that we think about safety, which is a key component in clinical trials, is predicated on this relatively, let's say, I would say outdated. If you consult the bioethics literature, it's actually pretty outdated. An outdated, very paternalistic model of a risk of inclusion that is not balanced by any sort of bioethical consideration about a risk of exclusion. And we haven't done that in an ecologically valid model. So, and by that I mean, we think about the risks of inclusion and exclusion. We need to consider that on the individual and the collective and then the larger societal levels. And now, a future of clinical trials that is equitable and efficient, I would say, in addition to being decentralized and virtualized, which are not quite the same thing, it needs to be democratized and ultimately decolonized. And I promise these are not just like word salad things that we say in Aspen. There are very, very concrete changes that we can make into the way that we design clinical trials to respect all of those ideals. But ultimately, we will only get to them. We will only get to, I think, a much more effective and equitable future of clinical trials if we do a better job of balancing the risks of not just of inclusion, but also the risks of exclusion. So arguably, you can suggest, what you can see is that many of the health inequities or disparities that we see, particularly with regard to maternal mortality, it isn't that necessarily care is getting worse in women of color. It's just that it's improving and accelerating much more quickly beginning in the 1980s, right when we started kind of changing our models of designing clinical research around this new population of convenience. And so what happened was on the collective level, these other groups were left out. There was a risk and there was a harm of exclusion from cutting-edge clinical research. So if we can do a better job of balancing those risks, and I think it's really hard to kind of put on our hat as trialists to think about like what kind of harm somebody might experience from not doing this study, then I think that opens the doors towards thinking much more carefully about like a truly decentralized trial. So I know that Amy, as an oncologist, says, maybe we can't do that all the time, but if we do a better job of balancing those risks, we might realize that in collecting a broader set of data, so instead of just data that is narrowly collected by the trial, thinking about increasingly technological-based or passive data collection measures, then you can really virtualize or decentralize a large percentage of that trial participation experience. You can use simulation studies to mitigate a lot of these adverse or side effects to the work that we're doing here. And so ultimately, you can create a very efficient, well-balanced, highly ethical research study. Now, this is the Aspen Ideas Festival. We're here to think big. We're not going to get there tomorrow. But if we start to think about these questions, again, from an individual person-led perspective, if we build in that context of being human in a clinical trial, then lots of solutions really just readily present themselves.
So I feel like we can think hard about where we want to go and what it should look like. From the regulatory perspective and regulators involved in that process, they can help us understand what good looks like and what they're going to find trustworthy and not. At the end of the day, the other pretty critical thing is we totally need a different kind of infrastructure, right? We actually need different capabilities in our ecosystem to build towards the clinical trials of the future. I personally voted with my feet. The reason why I left FDA and came to Verily was to start building those pieces, because what I could see is until you actually start building the pieces and putting them into play and saying, aha, here's what we can do, and regulators to say, yes, I trust that and I don't trust that, then we're really going to have hard time moving forward. So we need software in the clinical research workflow that helps real people understand for example, what they're consenting to and why, and how to participate for the long haul. We need to build towards making it easier to be able to bridge between research sites and decentralized capabilities. We need to build towards leveraging all available data, including data in our EHR and data that's intentionally collected for the clinical trial task. And we need to also have the level of compute power and algorithm development to pull that off. And so I think the what to do is actually not only to think about what we should be doing, but actually then try and put it into practice and then map backwards to say what worked and what didn't work and what do we need to continue to improve on.
Such a smart panel. All right, let's take some questions. Let's go right here.
Hi, I'm Jakub Hlavka, USC. I work in Alzheimer's research as well. I'm curious about the level of evidence or strength of evidence we need to get to approval. Partly with Aduhelm, we only had 18 months of data, and I do a lot of simulations on Alzheimer's disease. There's huge differences between projecting a treatment effect of 18 months and just assuming it lasts for 18 months versus lifelong effect, which actually has enormous economic consequences. So I'm just curious about how you think about designing trials to collect sufficient data, not just to meet the primary endpoint barely or at minimum, but to really collect evidence that we can then use to inform policymaking.
Yeah, so I'll try to respond quickly. I think that there are a couple of questions embedded in there. And I think with the question of Aduhelm and the aducanumab trials, which is a rant for another day, I promise. One of the questions is what kind of outcome means that we have successfully treated Alzheimer's disease? So we were going down this path of a long time where we said, oh, well, if you get rid of the amyloid, then there's some downstream things that will happen. Presumably, their cognition should be fine. We didn't see any of that in Aduhelm. We haven't seen that in a number of other trials that have ended before and after that. So the deepest question is, what is an outcome of interest? And frankly, this is where that patient-powered perspective might be most useful. So if you would talk to patients, they would say, okay, well, assuming that these biomarkers are high-fidelity markers of cognitive change, either now or in the future, like that's fine. But we're showing that that's not the case. So maybe let's just go back to measuring the thing that we intended to measure and having like a primary endpoint of cognitive change as being what was particularly relevant in there. Now that opens up a whole other can of worms. If you think about memory alone, it is really, really hard to measure that in a very fine-tuned way that will give you enough sort of variance to detect an effect after 9 months, 12 months, 18 months. So maybe we need to think, and this is where we get into my doctoral dissertation, maybe we need to think a little bit beyond sort of static measures of what we've got like in our CDR sum of boxes or the PAC-5, and think a little bit more broadly about some of those outcomes that, again, can be potentially informed by patient views. Now, more thoughtfully, though, we can think about designing a trial, maybe not for this round, but in the future, that takes advantage of new technologies, of doing sort of passive data collection. So the digital biomarker space in Alzheimer's research is completely exploding. You can overlay that onto like this traditional clinical research model, and then that can potentially give you the variance that you need. So if you don't have enough variance by looking at key time points with your primary endpoint of data that you have in a clinical trial, maybe overlaying some of these passive data collections, which will give you many more time points, which will introduce the variance that you need to identify a signal, is the way to go. So I think that there are a lot of different steps that we can take in this particular space. I won't get into issues of how you design a study that has 70% of 70% of participants with an E4 allele, even though black people there were only 19 individuals out of 3,300, even though black people tend to have the E4 allele at much higher rates than any other part of the population, there are still things that you need to do to kind of design for representation of the individuals who are most likely to develop the disease and make sure that your outcomes are powered for that presentation. But narrowly, we need to think very carefully about those endpoints, and if we can't get the data that we need using those traditional clinical research measures, potentially overlaying new and emergent biomarkers to help give us the variance that we need to potentially detect an effect.
Let's get in another question. Well, yeah, we'll come right here. How about you?
I'm a clinical trialist who led an NIH clinical trial group for 22 years, and one thing you didn't say, but I hope you do agree with, is that the trials need to be randomized and controlled.
Right.
And when we talk about inclusion, we need to be stratifying by the groups that we're looking at. Would
Maybe.
you buy that?
So I'm actually gonna have a couple of hot takes. Do trials need to be randomized and controlled? Randomized, sure, but do they need to be randomized at the level of the individual? Not necessarily. And that's how you get into thinking more broadly about the future effects of platform trials. And if you think about cohort multiple RCT designs, which randomize at the level of health systems in some cases, pragmatic trials. So randomization is important, but do we need to randomize at the level of the individual? Not always. I would say control is important. But we also need to think about what it is that we're controlling and making sure that we are adding important context so that there's not a huge gulf between what we think of as kind of traditional efficacy and then sort of real world effectiveness. So I think all of that's really important. But what I ultimately want to kind of narrow in on, and I know I can see from your faces and I can see from Amy that I'm touching on some tensions here, but I think that this is the right way to think about it. The traditional RCT design is designed very efficiently to produce a certain kind of evidence in a certain kind of time frame. We need to recognize that some trial designs need to move away from that traditional RCT space in favor of platform trials, in favor of N-of-1 designs, in favor of pragmatic designs to make sure that we have the breadth of evidence that we need to ensure that we're having an impact on the outcome of interest.
I would just hit on one really critical thing here, which is it's suddenly been in the last 3 or 4 years that randomization has become evil. And actually, randomization is pretty critical when we need to understand how a product compares to a standard or a placebo. We do have a lot of opportunity to innovate from there. And so, for example, randomizing initially for the first proportion of patients and then now starting to predict what the control arm is going to look like using real-world modeling studies. There's a lot of ways that we can continue to innovate in this space to reduce the need to randomize and allocate patients to one intervention or another once we have some of the basic pieces of understanding here. But one of the things that I think we've got to be careful about is that as we think about evidence generation in the future, randomization is a pretty critical part of dealing with equipoise, and that's really, I think, pretty critical.
Let's get to this question in the back.
Hi, thank you. As someone who works in an institution that's very heavily invested in big data comes from many sources. Most people have a cell phone.
Sir, could you hold that a little closer?
Most people have a cell phone, and we're interested in big data. And so the question becomes, with advocacy and not waiting for that patient advocacy, not waiting for that 10-year journey, can you comment about the pros, cons, dangers of crowdsourcing clinical trials? And can that be hypothesis testing?
So I think this is a really important area for us to spend more time doing work. Practically speaking, the ability to, there's different parts of what you've embedded in your question. So first of all, there's ability to think about using solutions that we carry with us every day, whether that's our phone or our watch, actually to help create critical endpoints and other digital measures that can now be embedded in our current regular clinical trials model. Then the second part of what I feel like you were asking is, can we also start to think about more crowdsourced mechanisms to now start understanding from communities, whole communities of patients, about what matters to them, about starting to understand critical initial hypotheses that then can be tested in more formalized ways. And certainly we've seen that, for example, in the breast cancer community and that kind of work. The last piece of what I felt like was embedded in your question is this critical question of data coming from many, many sources and now being put together in service of evidence generation. We've talked mostly about EHR and claims plus clinical trials data, but that story gets really wide really quickly, and we need to learn how to do it. Environmental data, social determinants of health, understanding personal preferences, and also the healthcare delivery system, and how do these things impact interventions and how they perform. We have a lot of work to do there. We need a lot of compute power to do it, but this is where the world should go.
We are just about out of time. I just wanted to thank our panelists for doing something that doesn't always happen at Aspen. We were talking about it before the panel. They went beneath that 30,000-foot level into the real details of what needs to change.
And we did not agree, which is nice.
And you did not agree. Another thing that rarely had a disagreement. So thank you so much. Really appreciate it, and enjoy Aspen.
Thanks.
All right.
It's great.
Thank you.